Hi, and welcome to this episode 8 of the rEView podcast. Today I've got a lovely guest, Leyla Esfandiari. Leyla, I'll let you introduce yourself.
Hi, Natalie. It's like so nice to be here, and thank you so much for this opportunity. As you mentioned, my name is Leyla Esfandiari, and I'm an associate professor of biomedical engineering at University of Cincinnati, Ohio, United States.
Great. So, can you tell me a little bit about what your lab does in terms of like context around EV research?
Sure. We started as a technology lab. Basically, we are working on developing nano, micro size devices for diagnostic application. And it's a little bit of a backstory I have for you here. When I first started my research lab as an independent I was interested in detecting microRNAs for early diagnostics of cancers, and I had a really lovely collaborator at School of Medicine at UC Cancer Institute. So as I was speaking to her and getting some feedback, she mentioned that besides microRNA that are very important, you kind of need to basically extract and isolate EVs if you could, and make devices that can actually purify those from biofluids, because the microRNAs are usually inside of these EVs. So that's how I got started getting introduced to EVs and the importance of EVs in diagnostics. And so we started basically modifying some of our devices that we had for nanopore sensing. We were interested in detecting sequences specific of nucleic acids, and based on this feedback, we basically changed the way we were looking at these devices and utilized them for trapping of EVs from biofluids in label-free manner. So my lab, I think I started here at UC in University of Cincinnati in 2015, almost 10 years ago. And I think in 2017, we started working on EVs and modifying and utilizing these devices for trapping exosomes. That time we were calling them exosomes. So in 2018, I actually filed for a patent of the device, and in 2023, we granted the US patent for the device for label-free isolation of EVs. So that's what we have started the lab, and basically we shift towards EVs. And now we are more and more interested in understanding if we could utilize them for regenerative medicine, tissue engineering, and so forth. So we are getting more into biology as we progress into our research.
Well, congratulations. That is an amazing achievement and
Thank
milestone
you.
to have that come through. And especially diagnostics, I have wondered for such a long time when we were going to start progressing because even myself in my PhD, I was doing like a biomarker study using EVs, and I just saw the field kind of producing all these papers that were looking at EVs as biomarkers, but never getting to the next step with an actual assay or device. So that's really amazing. So you said you kind of mentioned like in cancer and things. So what potential like applications do you see this being used for?
Yeah, sure. So now this device can be utilized for basically purifying EVs based on their unique biophysical properties, both size and their dielectric properties, means mainly their charge distribution and how they can polarize under the non-uniform electric field. So we're applying electric field to pull them, and it's the trapment of these EVs based on balance of three electrokinetic forces. I don't want to get more into details about and technical about the device, but so we utilize this to purify EVs from cell culture media, from different type of biofluids. We tried with blood, urine, and saliva so far, and we had different type of success in terms of purity and yield for different type of biofluids, because what we are utilizing, I mean, how the way that the device works, it depends on the permittivity of the media that these vesicles are in. So every biofluid has a little bit of a different properties. So that being said, we utilize the device for purification of the EVs. We actually have another module added to the system, and the patent is already filed and pending to characterize them based on their unique dielectric properties, as I mentioned. So we are interested in applying alternative current and looking at how they become polarized and how we could actually distinguish them based on their unique dielectric properties, which is one of their physical properties. So it's still a label-free manner. That actually was part of the main topic of my National Science Foundation Career Award in 2020. So going fast forward, after like setting up the device and kind of utilizing the device with collaborators and basically extracting EVs from plasma and other biofluids. And then we are more interested in understanding their functional role in regenerative medicine. We are mainly interested in peripheral nervous system, and we are looking at extraction of these EVs from Schwann cells that are very important in regenerating of the neurons in peripheral nervous system injuries. And that's very exciting because my lab is working with primary Schwann cells, and we are extracting EVs and looking into their different properties. Of course, we are doing some molecular characterization, looking at their transcriptomics, proteomics, and so forth. So we are moving forward with that project, and that's recently got funded by National Science Foundation Miro Award for the next 5 years. So I'm very excited about that. It's for earliest stage investigators. So very fortunate to have that in my lab. So we are moving forward with that. And there are some new projects that we are actually looking at tumor microenvironment and looking at the mechanobiology, basically mechanoelectrical feedback loop in tumor microenvironments, and looking at the EVs that are secreted from different stiffness of the spheroids that were made by these Cal27 cells. So we are expanding more into understanding the functional role, both in metastasis cancer, with collaborators, of course, and then on the other side, we are looking at their functional role for regenerative medicine,
Yeah,
mainly in nervous
wonderful.
system. So yeah,
That's amazing. Yeah. And congrats again. Double
thank
congratulations
you.
for your NSF. Yeah, that's amazing. So, I mean, it's really interesting because I come from a biology background, I guess, and
Yeah.
I'm intrigued by all the like the biotech or the bioengineering side of EV research. So, how did you make your way to EVs? What was your career path? And like you can go back as far as you need to go, like starting out with how you even got into science in the first place. But yeah, tell me the story.
Sure. I think science is very interesting. It's kind of like a mysterious path that you start in somewhere and then you end up somewhere else because you follow the unknown, basically. You want to solve the problems and understand the mysterious way that the nature works, basically. So I started as an engineer, did my undergraduate in California, Cal State Long Beach, in electrical engineering. And I was pretty successful as an undergraduate student, but I wanted to do something more in like application in biology, and I felt like electrical engineering was a little bit too dry for me. So I switched to my master, getting a master's at UC Irvine in biomedical engineering, and then I worked for a year in the industry, biomedical engineering company, and I felt like I'm not that much interested in being in the industry. So I wanted to go back and get my PhD in biomedical engineering. So I started my PhD at UCLA and started in the bioengineering department. And I met my PhD advisor when I took his class, and he was a great teacher and great mentor in my life. So I switched labs and joined his lab because I felt he was very hands-on and helpful. He was coming to the lab, training all the students. He was always available. So that was the kind of like I wanted to have someone that was there for me when I had questions and could help out in the lab. So I got trained by my PhD advisor in the lab. I was fortunate enough that he had time and resources to basically join us in the lab. And then after that, I started working on making a new devices for nanopore sensors as a nanopore sensors for detection of the nucleic acids. As I mentioned, that was my PhD. And I always wanted to become a professor and independent faculty. And I actually did not go for a postdoc. So I started applying the last year of my PhD for the faculty position, and my advisor thought I lost my mind or something. 'Cause he said, "You will never be able to get the position, tenure track faculty position." I said, "I will try." And of course, I applied for some postdoctoral positions too, and I had some offers as a postdoc, but University of Cincinnati at that time, they were looking for someone with biosensor expertise. So I was basically at the right place at the right time. So I applied, and then I started right after my PhD. I got my PhD in 2014, September 2014, and I started my independent faculty position in 2015, January. So I fast
Incredible.
tracked
That's
that.
amazing.
Yeah.
Yeah.
And then continue, like, of course, we are always engineers and we always like to kind of make gadgets or devices. But I think it's very important to kind of remember why we are doing what we are doing, just not to just make cool things that has no applications, because that could happen to engineers and they fall in love with their little devices. So I always try to keep current and be part of, actually, I maybe attend more kind of like biology, medicine sort of conferences, and I try to always have a connection with collaborators from the other side of the campus, which is the School of Medicine, Cincinnati Children's Hospital, to just remember what's needed in biology and just follow that and make devices or make tools to help out with medicine and understanding the biology in different type of disease or applications.
Yeah, definitely. I mean, you're right, and I think it's true of all scientists who are like highly creative, which I think is a lot of us. We get very carried away sometimes with the excitement of the concept and forget about some other practical real world applications. But yeah, that's I think it's such an interesting backstory to come from, yeah, electrical engineering and then
Civil
find your
engineering.
way. So what was the first exposure you had to EVs? Like where did that come up?
Yeah, so the first exposure, as I mentioned before, came from my collaborator at School of Medicine. She mentioned exosomes. And then I remember I was attending this I want to say it was biomarkers discovery, like the little smaller conference in Boston. And then when I met Jan Lodwall at in that conference, and he actually introduced me to the ISEV. He mentioned that the next one is going to be in Toronto. I think it was 2017 or 18. when I
So
first.
still fairly young at that point. Yeah.
Yes, yes. So I started joining and I learned that, oh, so much to learn and understand about EVs, and it's so complex, and the heterogeneity was fascinating. And so I kept coming every year to ISEV, except I think when we went remotely during pandemic.
Yeah.
I think I skipped that one, but other than that, I've been going to ISEP every year and learning a lot from biologists. Yeah.
Yeah, I mean, so obviously we met this year in
Yeah.
Australia, my home.
Yeah.
Um, and
Which was
yeah,
the best ISEP of all.
it was so good. Not biased at all, but
Yeah,
it
yeah,
was a
that's what
great
you do.
meeting. So yeah, I mean that's I always like to know how people first came across EVs. I mean, for me, I landed on it in my PhD, so that
Yeah,
was the first time I had ever heard the word
yeah,
extracellular vesicle, exosome. It was still at that
yeah,
time.
exosome. Yeah,
Yeah, so yeah, interesting to come across just from someone that you met that told you
yeah,
about them, and then that started off this whole journey for you.
yeah,
So we kind of like spoke briefly before the start of the podcast, and you said you're going to ISEVtech soon in
yes.
November. So are you presenting anything?
Yeah, I'm
Yep.
presenting, and it's going to be mainly on the device. So basically, because it's ISEPtech, so I'm going to
Yep.
present all features of the device and what we have done so far, and we're continuing utilizing them for different applications. So it's very interesting to see the results, and I have some interesting information to share.
Yeah, great. And I wasn't too sure like how it well, I haven't been to an ISEVtech, but it's only the second one that's ever been, right? So,
And this is my first time, so I
yeah.
missed the first first ISEPtech.
Yeah. So, Randy and Colin was saying that they kind of had stalls set up at the first one in Hawaii, and everyone could go around and test a sample and see. So, are you going to have anything like that set up with your device, or not this time?
This time, but maybe the next time we should do that, because that would be very interesting to actually, demo our work and devices. We need a little bit of, I must say, like one of the for characterization system, we need kind of like this powerful amplifier that we're measuring. So it might be a little bit hard to just,
Oh,
ship
you
the
can't.
stuff. Yes,
Yeah,
so
I can
it might be a little bit
imagine.
harder for, I
Can't
mean,
take the whole rig with you on the
academic, yeah,
road.
yeah, even it
They
might be
might
hard
look
to
at you
move.
strangely on
Yeah,
the
yeah,
plane when you
yeah, exactly. So, but I'm sure like folks from the industry, tech companies, I'm sure they can just transfer, all sorts of equipment for different
I know.
conferences.
I mean, I see them setting up at the conferences and I wonder
Yeah,
like how did they get all that stuff
yeah,
there with them? What kind of planning does that take? So
yeah, yeah.
yeah, so since are you one of the only people working with EVs at University of Cincinnati, or are there a few groups, or have you started fostering
Yeah,
interest in EVs?
yeah, interesting. So in the beginning, when I first started working on EVs, I think there were a few other groups at UC and Children's Hospital that were working on EVs, and they were mostly coming from School of Medicine and biology side of. So in terms of technology, I think I was the only group, and I'm still thinking, mostly I'm the only group, but there are some other faculty that's starting their labs and they're going to utilize their devices, mainly sensors for EV application. But yeah, we started small, so not that many folks were working on it, and now we are expanding because at University of Cincinnati we are starting or building a new nanobioscience institute, and I'm one of the executive committees in that new center. So it's very heartwarming because we are going to have kind of branch or program dedicated to EVs and nanomedicine. So I'm planning to lead that. And as I was looking to see who's working on EVs to kind of gather everyone together, I noticed like there are a lot of folks right now at UC and in the region, basically Cincinnati Children's Hospital, VA hospital, that are interested in EV work. So we are expanding definitely, and it's very nice to see that we can do more collaborations, better exchange of information and so forth. So we're excited.
Yeah, very. And I think like this is kind of I've seen it happen at a few different universities that this is becoming a thing. Like EV cores are being
Mhm.
established or
Yeah,
nanomedicine, nanotech, like that kind of core is happening and
yeah.
EVs get roped in with that. So
Mhm.
yeah, that's it's exciting times for sure. So do you think like what do you see happening in the field over the next kind of 5 to 10 years? What's going to be the next most exciting thing?
Because there's still so much to learn about EVs. I think it's going to grow and it's going to become more and more popular as has been happening for past few years, I want to say. But in terms of technology, I think we are going to have both bulk extraction and characterization of the EVs, and then we're going to go down, of course, to the single EV resolution. So I think both technologies are important because we have such a heterogeneous, population of EVs. So it's good to have both and work on both systems. And I think it's going to be more and more interest towards label-free manner because it's faster. It's much more convenient. So whoever comes in my lab basically can utilize the device, and it's not so hard to learn. So I'm so happy about that. You don't have to go through labeling and so forth. Um, and I think in terms of single molecule, single EV, basically extraction or detection, we had great progress in the past few years, and I think we're going to continue moving forward. In terms of, I think we're going to utilize these EVs in terms of more tissue engineering, regenerative medicine, utilize them and understand their real functional role in aging and both, of course, diagnostics as well. So it's very exciting. I hope that we can continue working on it and understand the mysterious functional properties of these little vesicles.
Yeah, I mean, there's so much still mechanistically to
Yeah,
understand about even the way they're taken up. And I think
yeah,
like targeting, it seems to be such a
yeah,
huge area of interest, but also something that is so challenging as well. Like
yeah.
trying to improve the efficiency of the
Mhm, mhm,
delivery and the uptake. It seems no matter what happens, you can put in like 1 by 10 to the 10 EVs and only 1% might get taken up, which is still like in the figures of 10 to the 10, but not quite what you'd want if
yeah.
you're really trying to get something so specific. So, like from the engineering point of view, you think that this is like potentially possible we can start reaching more like a 80-90% efficiency?
Understand. Of course, markers on the EVs and how the uptake's happening. It's very fascinating, and we can continue that road. But I think in terms of looking at it as engineering point of view, understanding the true physical properties of these EVs, their thinking about the not the softness, but their
Like the
physical.
fluidity
Yeah,
and
not the
yeah.
fluidity. I want to say they're what's the word I'm looking for? How they fuse to the membrane. Come on,
Mm.
biophysical properties. Let's continue with the biophysical
Yeah.
properties.
Like
So
I always found like the EV surfaceome and the like the corona and all of these molecules that we have been trying to strip away for so long
yeah,
now seems like they're so important. So
yeah, yeah,
literally like just what they're being carried in, what the fluid properties are, and how it interacts, the EVs interact with those other molecules. I find that absolutely fascinating. And like
Yeah.
I see they're having the EV biomolecular corona workshop. I think it's in Italy. It's coming up soon. The abstract
Yeah.
deadline is 21st of October. So by the time this podcast is released, it would have
Mm,
been passed. But
yeah.
if I had an abstract to submit, I would totally want to go to that just because I think it's so interesting.
Very
But
interesting. Yeah.
not, yeah, unfortunately
No,
not
there's a
working on
lot
that.
going on, and we actually started seeing some very interesting ion channels on EVs.
Oh,
So this is
really?
a real new
That's
discovery. So
surprising.
it's
Yeah.
coming up. Yeah, it's and we've been validating this with three, four different techniques, doing westerns multiple times, and we seen some of the ion channels that we were very surprised that they were on EV. So
Now, is
yeah,
this EVs from a specific type of fluid or
So
cell
they're
culture
coming from,
media?
yeah, so they're coming from these Cal-27 cells that are in this stiffer tumor microenvironment. So they're feeling the physical, pressure or basically forces, mechanobiology forces that they feel, the cells feeling, and then they're secreted EVs from those cells from the tumor microenvironment. And interestingly, we saw piezo 1 ion channels on these EVs. And we actually filed for another patent because we were just so interested. I mean, it was so shocking and interesting at the same time to see that they carry ion channels and they can actually help modulating mechanoelectrical basically communication between cells. I have no idea how they're gonna,
That's
yeah.
crazy.
So yeah,
I did not. Yeah, that's
this is
amazing.
one of the projects that's going on, and I think the preprint is gonna come out soon. My PhD student Molly Sheth,
Yes. Yep.
that's her work, and she's right now writing it up, and she has a lot of success, and she's going to be great in the future working
That's
on
so exciting.
these.
So you said PSO1 ion channel. So
Yes.
I admit I don't know what that ion channel is. So what's the full does it have a full name or and what
No,
does it
no, it's
do?
a piezo 1 ion channel, and
Mhm.
so basically these ion channels help when you're applying mechanical forces on the membrane of the cells. These ion channels open up, and then it's the calcium will basically flow through. The Nobel Prize was awarded for discovery of piezo 1 ion channels, which are the mechanosensitive ion channels. And we could actually see them on these EVs that are under mechanical tension in a tumor microenvironment. So this is a very interesting observation or discovery for us. So we keep going and we're gonna try to understand their functional role, the pathway, and yeah, so it's a lot to discover and learn and understand still.
Yeah,
But
I mean that's quite
yeah,
the finding that yeah, because I
yeah,
haven't seen so far. Maybe and obviously I don't know every
yeah.
single EV paper that comes out, but clearly this seems to be like new information.
Yeah, and we haven't
But
published it. We noticed it almost. I think it was we could confirm it last April, and the interesting part was that because we were seeing the trace of these piezo ion channels on EVs, and I was just so surprised. So I actually sent out these EVs to a collaborator of mine, a School of Medicine, and asked them to run a Western just independently, and said, "Can you guys, see anything?" So I wanted to make sure that we are not biased about it, and they could actually show that, yeah, Western shows that there's a trace of piezo 1 ion channels on these EVs. Continue working on it. We done flow cytometry. we done ONI imaging to just validate all of that, and we could actually see that. So it's pretty interesting. I don't know where we're
Yeah.
going with this, but
No, I mean, no, it is very interesting. And like, I
yeah.
wonder, like, what happens when you block the receptors? Like, how, yes, like,
So now we have so you actually can block them with basically drugs, but then at the same time we as soon as we notice them, we actually try to basically clone these cells with knockout piezo one ion
yeah,
channels
yeah.
in it. So we have a knockout, and now we are doing experiments on those to see what will happen to the EVs. So yeah,
Wow.
so we are working on it, but it's a long
Wow.
way to go. But it's interesting observation and discovery. So we are excited, very exciting time in the lab.
Yeah, definitely.
Yeah.
I'm excited.
Yeah, thank you.
And you know, I mean, I love EVs, but I love these
Me
new
too.
things that we find out about them.
Yeah, yeah,
Seems I say seemingly all the time. It depends who's looking, you know, like because you're coming from this alternative perspective, not
mm,
just biology background, but engineering background. I just think, yeah, this is where the real discoveries are made because someone with a
thank you.
different point of view and
Mhm.
different skills
Yeah.
can come in with a different approach. So yeah, that's
I
really
think
cool.
the fact that we discovered this was because we were interested in mechano-electrical feedback loop in tumor microenvironments, and Molly was interested in piezo one ion channels on the cells. So she was like working with the cells, and then one day when she was basically doing Western for the EVs that was secreted from these to just look at the, normal markers, CD, I mean, tetraspanning markers. Then she just had this piezo one as well, so just see what she would get. And surprisingly, we noticed like, oh, you have piezo one. I'm like, oh, okay, probably it's not true. So let's maybe try again, and then try again, and try again. And 6 months later or 7 months later, we still have we see the trace of piezo one ion channels on EVs. So I wonder how many more ion channels can be embedded on extracellular vesicles. So yeah, very interesting.
Yeah, that's great.
Yeah.
So yeah, I mean, this might be a nice time to kind of wrap things up, but um, I guess, yeah, is there anything else you want to talk about before we finish? Like anything about your work or upcoming things or anything at all?
I think yeah, just I think the field of EVs is just so interesting, and we're gonna keep continue working on both developing some devices and also making these biomimetic scaffolds. We are getting more into tissue engineering and regenerative medicine. So we're actually having these piezoelectric nanofibers that we can stimulate the cells with electric field, applying ultrasound to them, and then looking at the secretion of the EVs as well. So we are basically interested in understanding EV's functional role or secretion or yeah, in regenerative medicine and both diagnostics and therapeutics in a kind of like non-traditional fashion. So we are taking some chances to understand and study EVs. And in the future, I think we will expand and continue working on them and maybe make new devices as well. So it's just a very exciting time in our lab and in our group. So yeah,
Oh, well, thank you so
yeah.
much
Thank
for
you so
coming on the
much.
podcast, Layla. It's been an
Thank you.
absolute pleasure.
Thank
I
you.
hope to run into you at future conferences.
Yeah, you're not coming to ISEVxTech?
Unfortunately, no, I have no reason to go. You know, I was
I see.
hoping to maybe make it to ISO 2025. Not sure on that yet. We'll see.
Yeah,
But
it's a long travel from the US.
I know. So at least now that I'm in the US, we'll think about yeah, national ones that I can get to
Yeah,
instead. But
yeah.
yeah. But thank you once
Thank you.
again.
Thank you
Appreciate
so
it.
much.
And we'll say bye to our listeners. Goodbye, everyone.